Today, we delve into the fascinating world of biotechnology and the groundbreaking realm of CRISPR-based therapies. As we embark on this scientific journey, we'll unravel the mysteries behind gene editing, explore the potential of CRISPR technology, and discover how it's revolutionizing the landscape of medicine and beyond. What is CRISPR? CRISPR, pronounced CRISPR, stands for clustered regularly interspaced short palindromic repeats, which are the hallmark of a bacterial defense system that forms the basis for CRISPR-Cas9 genome editing technology. In the field of genome engineering, the term CRISPR or CRISPR-Cas9 is often used loosely to refer to the various CRISPR-Cas9 and CPF1 and other systems that can be programmed to target specific stretches of genetic code and to edit DNA at precise locations, as well as for other purposes, such as for new diagnostic tools. CRISPR technology offers the promise to cure any human genetic disease with gene editing. CRISPR/Cas9 was first used as a gene editing tool in 2012. CRISPR is short for clustered regularly interspaced short palindromic repeats. The term makes reference to a series of repetitive patterns found in the DNA of bacteria that form the basis of a primitive immune system, defending them from viral invaders by cutting their DNA. Using this natural process as a basis, scientists developed a gene editing tool called CRISPR/Cas that can cut a specific DNA sequence by simply providing it with an RNA template of the target sequence. This allows scientists to add, delete, or replace elements within the target DNA sequence. This system represented a big leap from previous gene editing technologies, which required designing and making a custom DNA cutting enzyme for each target sequence rather than simply providing an RNA guide, which is much simpler to synthesize. CRISPR gene editing has already changed the way scientists do research, allowing a wide range of applications across multiple fields. But the technology could also hold great potential as a treatment for human diseases. In theory, CRISPR could let us edit any genetic mutation at will to cure any disease with a genetic origin. In practice, however, CRISPR is still in the beginning stages of its therapeutic development. Here is a list of some of the first diseases that scientists are tackling using CRISPR technology, testing its possibilities and limits as a medical tool. Cancer. Cancer is a complex, multifactorial disease, and a cure remains elusive. There are hundreds of different types of cancer, each with a unique mutation signature. CRISPR technology is a game-changer for cancer research and treatment as it can be used for many things, including screening for cancer drivers, identifying genes and proteins that can be targeted by cancer drugs, cancer diagnostics, and as a treatment. China has spearheaded the first in-human clinical trials using CRISPR as a cancer treatment. The gene editing technology was used to remove the gene that encodes for a protein called PD-1 that some tumor cells can bind to block the immune response against cancer. CRISPR technology has also been applied to improve the efficacy and safety profiles of cancer immunotherapy, such as CAR T-cell and natural killer cell therapies. In the US, CRISPR Therapeutics is one of the leading companies in this space, developing off-the-shelf, gene-edited T-cell therapies using CRISPR. Despite these milestones, CRISPR technology still has limitations, including variable efficiency in the genome editing process and off-target effects. Some experts have recommended that the long-term safety of the approach remain under review. Others have suggested using more precise gene editing approaches such as base editing. Blood disorders. The blood disorders beta thalassemia and sickle cell disease, which affect oxygen transport in the blood, are the target of a CRISPR treatment being developed by CRISPR Therapeutics and its partner Vertex Pharmaceuticals. This exa-cell therapy, which is currently being tested in 5 clinical trials, consists of harvesting bone marrow stem cells from the patients and using CRISPR technology ex vivo to make them produce fetal hemoglobin. This is a natural form of the oxygen-carrying protein that binds oxygen much better than the conventional adult form. The modified cells are then reinfused into the patient. In September, exa-cell was granted a rolling review by the US FDA as a potential one-time treatment for sickle cell disease and transfusion-dependent beta thalassemia. This follows the granting of Regenerative Medicine Advanced Therapy, Fast Track, Orphan Drug and Rare Pediatric Disease designations. If approved, exa-cell will become the first CRISPR therapy to achieve regulatory approval for a genetic disease. Vertex will also submit a biologics licensing application for exa-cell in November and a marketing application to the European Medicines Agency by the end of the year. Hemophilia is another blood disorder that CRISPR technology could tackle, although development is still at the preclinical stage. Blindness. Many hereditary forms of blindness are caused by a specific genetic mutation, making it easy to use CRISPR/Cas9 to treat it by targeting and modifying a single gene. In addition, the activity of the immune system is limited in the eye, which can circumvent any problems related to the body rejecting the treatment. The treatment aims to use CRISPR to restore the function of light-sensitive cells before losing sight completely by fixing the most common genetic mutation behind the disease. In 2020, the company started a phase 1/2 trial, which was the first trial to test an in vivo CRISPR treatment. This is when gene editing is performed directly inside the patient's body rather than on cells extracted from their body. AIDS. There are several ways CRISPR could help us in the fight against AIDS. One is using CRISPR to cut the viral DNA that the HIV virus inserts within the DNA of immune cells. This approach could be used to attack the virus in its hidden, inactive form, which is what makes it impossible for most therapies to completely get rid of the virus. In September, the first ever individual with HIV was dosed with a CRISPR-based gene editing therapy in a phase 1/2 trial led by Excision Biotherapeutics and researchers at the Lewis Katz School of Medicine at Temple University in Philadelphia. Another approach could make us resistant to HIV infections. Certain individuals are born with a natural resistance to HIV thanks to a mutation in a gene known as CCR5, which encodes for a protein on the surface of immune cells that HIV uses as an entry point to infect the cells. The mutation changes the structure of the protein so that the virus is no longer able to bind to it. This approach was used in a very controversial case in China in 2018, where human embryos were genetically edited to make them resistant to HIV infections. The experiment caused outrage among the scientific community, with some studies pointing out that the CRISPR babies might be at a higher risk of dying younger. The general consensus seems to be that more research is needed before this approach can be used in humans, especially as recent studies have pointed out this practice can have high risk of unintended genetic edits in embryos. Cystic fibrosis. Cystic fibrosis is a genetic disease that causes severe respiratory problems. Although there are treatments available to deal with the symptoms, the life expectancy is only around 40 years. Cystic fibrosis can be caused by multiple different mutations in the target gene CFTR, over 700 of which have been identified, making it difficult to develop a drug for each mutation. With CRISPR technology, mutations that cause cystic fibrosis can be individually edited. In 2020, researchers in the Netherlands used base editing to repair CFTR mutations in vitro in the cells of people with cystic fibrosis without creating damage elsewhere in their genetic code. In addition, companies such as Vertex Pharmaceuticals and CRISPR Therapeutics have plans to develop treatments for cystic fibrosis using CRISPR systems. However, these therapies still remain in the research phase. Muscular dystrophy. Duchenne's muscular dystrophy is caused by mutations in the DMD gene, which encodes for a protein necessary for the contraction of muscles. Children born with this disease suffer progressive muscle degeneration, and existing treatments are limited to a fraction of patients with the condition. Research in mice has shown CRISPR technology could be used to fix the multiple genetic mutations behind the disease. In 2018, a group of researchers in the US used CRISPR to cut at 12 strategic mutation hotspots covering the majority of the estimated 3,000 different mutations that cause this muscular disease. A company called Exonics Therapeutics was spun out to further develop this approach. A year later, it was acquired by Vertex Pharmaceuticals for approximately $1 billion to accelerate drug development for this disorder. If you enjoyed today's video, give it a thumbs up, share it with your fellow science enthusiasts, and don't forget to comment down below with your thoughts and questions. Make sure to subscribe to our channel for more mind-blowing content on the latest advancements in science and technology. Remember, the future is now, and we're here to unravel its mysteries together.